In this segment, I'm going to talk about the helminths. So helminths can be classified as nematodes or platyhelminths, so roundworms or flatworms. And then there's two subcategories of the flatworms. Cestodes are tapeworms, and trematodes are flukes. You should know the common name and scientific name for each one of these. So sources for human infection with worms include food, soil, water, and infected animals. The routes of infection include oral intake or penetration of unbroken skin. Humans are the definitive host for many species. So instead of being an end host or a terminal host, they are the definitive host, which means that the parasite was evolved to infect a person. Animals or insect vectors can also serve as reservoirs. And then here's a list or a table of some common roundworm infections, flatworms, trematodes, and cestodes. So you can take a look at these as you'd like. You are only responsible for knowing the specific pathogens that I'm going to discuss in this lecture. So how do we classify and identify them? So similar to identifying protozoans, we're going to identify many of these through stool samples, but there's a lot of other types of samples that we can take. Sometimes you can see the worm directly in an eyeball sticking out through the skin, underneath the skin. You can do a skin snip where you can see it in skin tissue. Sometimes you need a liver biopsy to see flukes. You would need to look at stool samples for eggs or fragments of the worms or entire worms. Sometimes people cough up the worms. So there's lots of different ways to investigate the worms. Classification criteria include the shape, size, the degree of development of organs, the presence of hooks, suckers, or other special structures on the whole worm, the mode of reproduction, the kind of host that it is in, and the appearance of eggs and larvae. Identification can be microscopic, so you can detect the adult worm, larvae, or eggs, and they often have distinct shapes or external and internal structures. Occasionally cultured to verify all life stages. So this is a picture of some eggs. You can see how different the eggs look. Now, some of them are kind of similar, but there's some unique features of each one of these eggs. When you look in a stool sample, these eggs are pretty obvious. They stand out pretty well. Sometimes they're very close, like if you can see these, like Clinorchus sinensis, looks like this one, but you can tell slight differences from them. So measuring the size of the egg, considering the host, the symptoms, and what part of the world the person was visiting when they got this, that all gives you clues as to the identification of the parasite. About 50 species of helminths parasitize humans. They're distributed in all areas of the world that support human life, and they may be geographically restricted, and there's a higher incidence in tropical areas. Yearly estimate of worldwide cases numbers is in the billions. Conservative estimate of 50 million infections in North America. Sometimes people get these infections and don't have any symptoms and end up passing them on their own. Other people will not have symptoms and be asymptomatic carriers for a long period of time before developing symptoms. Humans evolved in constant presence of helminths. Only recently have humans evolved to a helminth-free existence. So now we use drugs to eliminate our bodies of helminths. The absence of helminth infection may contribute to autoimmunity and allergy. People that have worms are less likely to have allergies. Now, this isn't saying that everyone should go get worms and go get parasites inside of them. It's just an interesting thing to notice, that when you're infected with a parasitic worm, you're less likely to have allergies. So now I'm going to talk about 4 specific species of worm that you need to know for this course. So the first one is Ascaris lumbricoides. This is a nematode, so it's a roundworm, and it's a pretty common roundworm. Transmission is ingestion of eggs from the soil. So what happens is you're going to eat the egg, and the egg hatches open in your intestine, and the larvae emerge from the egg. The larvae will actually escape your intestine, go into your bloodstream, and go to your lungs. And while in the lungs, they're going to develop into adult-sized worms. At night, while you're sleeping, these worms will crawl up from your trachea, from your lungs and go back down. You kind of swallow them inadvertently in your sleep, and they'll go back down into your intestines. In the intestines, they're going to grow and multiply and release more eggs. So they started out in the intestines, and then they had to do that gross lung thing just to come back to your intestines. Usually you don't notice that they're in your lungs, and there's usually no symptoms associated with that. The symptoms start when they're in your intestines. You can have hundreds of worms in your intestines, so they can multiply, and you can have hundreds and hundreds of worms. These are going to, or have the potential to cause potbelly, so you can see a distended abdomen from the presence of the worms. It's easily treated with drugs, but symptoms include weight loss, fatigue, irritability, nausea, and diarrhea. Tinea solium is a cestode, so it's a tapeworm, and this is the pork tapeworm. So transmission is eating undercooked pork. The live worm in the meat hooks into the intestines using its scolex, which is a mouth part. If the human ingests the eggs, though, the larvae will migrate through the tissues and can enter into the brain. So if you eat the worm, it's not as bad as if you eat the eggs. If you eat the eggs, they'll hatch. Those larvae will travel all over your body, and they can end up in the brain. The drugs can kill the worms, but they can't fix the damage. So this is a picture of the pork tapeworm in the brain. You can see all of those cysts are not normal. This may be why the Jewish religion says that pork is unclean meat. Since we're talking about cestodes, I want to just click back and go through the typical life cycle of the cestode. And there's a picture of one way back when. Here we go. So a cestode, a tapeworm, usually when you get infected with a standard tapeworm that lives in your intestines, it's going to attach to you using its mouth part, its scolex, and then it's going to grow, and it can grow almost the entire length of your small intestine. And here you can see how long these tapeworms can become. Usually only one worm will infect your intestines at a time. They do not like to compete, and they won't be comfortable having multiple worms colonize. Each one of these fragments, these proglottids, can break off to become a new worm. They also release eggs, and they have those egg structures inside of them, so they can spread eggs and they can actually break off themselves to become a new worm. Funny story about my cat. So I was eating breakfast one morning, and my cat jumped up on the table next to me, and this was several years back, but all of a sudden this little white thing fell out of her butt. And I was watching it for a while, and it kind of looked like an inchworm. It was like moving around and squiggling around. So I put it in a baggie, and I was thinking to myself, what could this possibly be? Was this attached to the outside of her, or was it actually like a worm that came out of her? So a couple hours later, I looked at the worm in the bag, and it had shriveled up into a yellow, what looked like a grain of rice. So it was about the size of a grain of rice, and it was yellow. When it did that, I knew for sure that it was a tapeworm. What tapeworms look like when they dry up and they're on the outside and they're trying to transmit from one organism to the next is they look like a grain of rice. So if you're looking at the fur around the anus of an animal, you can actually see them stuck to the fur. Now, people usually don't have furry butts, so you won't see this a lot in a person, but it's just something to notice on an animal. If you see that, that's a pretty common sign of a tapeworm infection. And these can be easily treated with an anti-Helminth drug. There are some people that use tapeworms as a weight loss plan. So they will consume a tapeworm, the tapeworm will grow inside of them, eat their nutrients, they'll lose a whole bunch of weight. But the problem is the inflammatory reactions that happen. So it causes a lot of inflammation, a lot of discomfort, and that inflammation can result in you end up losing your intestine or part of your intestine. So it can cause pretty severe diarrhea and discomfort. It is definitely not the way to diet, but some people think that that's appropriate. And then Schistosoma hematobium. So this is a trematode or a flatworm, a fluke. Transmission is snails hatch these larvae. The larvae escape and penetrate into the host's skin. So if you go bathing in snail-infested water, the snails are going to consume the eggs of the schistosoma, and those eggs are going to hatch inside of the snail and turn into larvae. the larvae are going to leave the snail. So the snail is an intermediate host. And when the larvae are in the water and you are walking in the water, those larvae can go right through your skin. When they get through your skin, it feels like a little bit of pins and needles. It doesn't feel like harsh pain or anything. It's just a little bit itchy. Sometimes there's inflammation around the area, but you usually don't notice it. Those will then go through your bloodstream, and they're going to coat themselves with your blood cells, with proteins from your blood cells, so it can actually hide from your immune system. Adult flukes will attach to blood vessels and migrate through the bloodstream. They can eventually get into your liver, they can go into your urinary bladder, and when they get into those areas, various species of schistosoma are going to cause liver failure or even bladder cancer. And that bladder cancer can come out of nowhere, and usually you don't know that you're infected. So if you're in endemic areas, you should definitely not bathe or walk in snail-infested water. Prevention and treatment, again, don't stand or bathe in snail-infested waters in endemic areas. And this one is treated with drugs. The problem is, by the time you usually diagnose this, it's because you went to the doctor with bladder cancer or liver failure or liver cirrhosis. So here's a picture of schistosoma hematobium in tissue. This is a good table that shows you some of the differences between eukaryotes, so protozoa, fungi, algae, and helminths, and then humans. I recommend taking a look at this table just to familiarize yourself with what's found in each one of these organisms.